Welcome. Today, we're diving into the work of Dr. S.K. Singh, focusing on dissolution method development and IVIVC studies. We'll explore how these techniques help bridge lab results with real-world drug behavior, making treatments safer and more effective. Let's get started. Let's start by looking at why dissolution testing matters. It measures how quickly and how much of a drug is released from its dosage form into a liquid under controlled conditions. This tells us a lot, from predicting how the drug will behave inside the body to making sure every batch is consistent. It also helps with developing new formulations, setting product standards, and navigating regulatory hurdles. Dissolution testing applies to many forms: tablets, capsules, modified-release products, suspensions, and even transdermal systems. Importantly, it's recognized as a critical quality attribute in modern pharmaceutical design. When we talk about global regulatory expectations, we're looking at a framework built on well-established guidelines. The ICH guidelines set the foundation, covering everything from pharmaceutical development to quality risk management and even analytical procedure development. Alongside these, regulatory references like USB standards and guidance from the FDA, EMA, and WHO provide detailed direction. Together, these aim to ensure product quality, predict how a drug behaves in the body, and establish methods that truly distinguish product performance. It's a comprehensive system designed to keep medicines safe and effective worldwide. Let's talk about the principle of dissolution, the process where drug molecules break away from their dosage form and enter the surrounding fluid. This step is crucial because it affects how the drug becomes available in the body. To study this, we use dissolution testing. It's not just about running a test. The conditions matter a lot. We carefully control the type of apparatus, how fast it stirs, what the medium is made of, and the temperature. Each factor plays a role in how accurately we can predict drug behavior. Let's talk about the USB dissolution apparatus, the tools we use to test how drugs release their active ingredients. Apparatus I, the basket, is great for capsules and floating dosage forms. Apparatus II, the paddle, is the most common, ideal for immediate-release tablets and capsules. Then there's apparatus III, the reciprocating cylinder, designed for modified-release products. Lastly, apparatus IV, the flow-through cell, works well for poorly soluble drugs and extended-release formulations. Choosing the right one depends on the dosage form and how the drug is meant to release. When developing a dissolution method, we start by evaluating the API's key properties, things like solubility, pKa, and its biopharmaceutics classification system category. Next, we select the right dissolution medium, choosing from options like 0.1N hydrochloric acid or buffers at pH 4.5 and 6.8. Then, we pick the appropriate apparatus and fine-tune the agitation speed. Finally, we set the sampling intervals, the ultimate goal: to create a method that's sensitive enough to detect any changes in the formulation. Now, let's look at an example of sync condition assessment. This process helps us understand how well a system handles its workload before it actually fails. By testing the limits and observing where performance drops, we can identify weak points early on. It's like stress testing a bridge. We want to know exactly when and how things might break, so we can reinforce them ahead of time. This example will show us how that works in practice. Here's an example of dissolution data, showing how a drug releases over time. At 5 minutes, about a quarter of the drug is released. By 10 minutes, we're halfway there. 15 minutes gets us to 75%. After half an hour, over 90% has dissolved. And by 45 minutes, nearly the entire dose is released. This kind of data helps us understand how quickly a medication becomes available in the body. Now, let's look at the similarity factor, or F2. This calculation helps us compare two sets of data, usually how closely one profile matches another over time. It's a key tool when we want to see if two things behave alike or if a new version stays consistent with the original. Understanding how to calculate and interpret this factor gives us a clearer picture of similarity, and that's crucial for making informed decisions. Let's start by exploring what IVIVC actually means. It's a predictive mathematical relationship, a way to connect how a drug dissolves in the lab, in vitro, with how it's absorbed inside the body, in vivo. This link is powerful. It can reduce the need for extensive bioequivalence studies, speed up product development, and offer more flexibility with regulators. Plus, it supports optimizing formulations. Both the FDA and EMA provide clear guidance on IVIVC, making it a trusted tool in drug development. Let's take a closer look at how we classify IVIVC, that's in vitro-in vivo correlation. It's all about linking what we see in the lab, like drug release from a tablet, to what actually happens inside the body. There are different levels or types of correlation, each with its own purpose and complexity. Understanding these classifications helps us predict how a drug will perform in real life, based on lab tests. It's a crucial step in drug development and quality control. Here, we see how absorption and dissolution progress over time in this level, a IVIVC example. After just 1 hour, around 18 to 20% is absorbed or dissolved. By 2 hours, that jumps to about 42 to 45%. At 4 hours, we're looking at roughly 3/4 absorbed or dissolved. And by 8 hours, the process is nearly complete, reaching over 90%. This data helps us understand how the drug behaves in the body, linking dissolution to absorption directly. Now let's talk about prediction error calculation. This is where we measure the difference between what we expected to happen and what actually did. It's a crucial step because it tells us how well our model or system is performing. The smaller the error, the better our predictions. But when the error is large, it signals that we need to adjust our approach, learn more, and improve accuracy. This process keeps us moving forward. Let's talk about how IVIVC plays a crucial role in regulatory submissions. It supports a range of applications, from SUPAC changes like manufacturing site moves, equipment swaps, or process tweaks, to formulation optimization and setting product specifications. One of the biggest wins, it can help waive the need for additional bioequivalence studies. That means less time and money spent without compromising on product quality or safety. It's a powerful tool for streamlining development and meeting regulatory expectations. When it comes to developing a dissolution method, we need to dig deep into the properties of the active ingredient. Choosing the right media and apparatus is just as crucial. We also have to make sure the method can clearly distinguish differences and validate it thoroughly. On the other hand, a solid IVIVC model lets us predict how the drug behaves in the body, easing regulatory hurdles and supporting product management throughout its life cycle. Ultimately, success hinges on sound methods, proper validation, strong models, and ongoing monitoring. That brings us to the end. If you need any more support or have questions, please don't hesitate to reach out. Thank you for your time and attention. It's been a pleasure sharing this with you.